T. Okay. So okay. Okay. So we're going to start these participants. All right. So as a summary, last time we talked about, we're still in our 3-2-1 hematopoiesis. If you look at the previous lecture where we have our course outlined, we have hematopoiesis as the first lecture to done. We are still on the series of hematopoiesis. So last time we said that hematopoiesis is a continuous and regulated process of blood cell production, and it includes cell renewal, proliferation, differentiation, and maturation of cells. That the process of hematopoiesis results in the formation, development, and specialization of all functional blood cells. We also say that during fetal development, hematopoiesis progresses through the mesoblastic phase, hepatic phase, medullary phase, and some organs function at some point in the process. Okay, some of these organs include the liver, the spleen, the lymph node, the thymus, and the bone marrow. Okay, so that the bone marrow is primarily the primary site of hematopoiesis at birth and throughout life. Okay, in certain situations, the blood cell production may occur outside of the bone marrow, and it is termed the extramedullary hematopoiesis. So, and we also have the hematopoietic stem cell. We talked about it, that is capable of self-renewal. We said that they are pluripotent, have the capacity to differentiate into different types of blood cells. Okay. Okay. And we also say that as the cells mature, they develop certain morphological characteristics that allow them to be recognized. We talked about cytokines, how they contribute to this process of hematopoiesis. And we say that the cells of the body are made up of, there are different types of cells in the body. We have the erythrocytes, we have the white blood cells, which can be broken down into 5 major types, and we have the platelets. So we are going to take these blood cells one by one and look at their production, and that is why we are starting today with erythropoiesis. Going to start with erythropoiesis. So in this lecture, you should be able to discuss the differences between reticulum of reticulocytes and punctate basophilia, be able to describe what is erythropoiesis, okay? Describe describe erythropoiesis as a process of development of red blood cells. Okay, so the red cells, or the erythrocytes, which we are going to talk about its production today, the red blood cells or erythrocytes, they provide a classic example of the biologic principle that cells have specialized functions. That cells have specialized functions, and that their structure are specific for those functions. Okay, each cell in the body have a specialized function, as we are going to learn. There is no other cell in the body that has the capacity to transport oxygen in the body or remove carbon dioxide except the red blood cells. Then the white blood cells are involved in immune response, and they also have their different specialization. That is what we mean when we say that each cell of the body have specialized functions. and that their structures are specific for those functions. The erythrocyte, take for example, has one true function, and that is to carry oxygen from the lungs to the tissues where the oxygen is released. This is accomplished by the attachment of oxygen to hemoglobin, the major cytoplasmic component of mature red blood cells. The role of the red blood cell in returning carbon dioxide to the lungs and buffering the pH of the the role of the red blood cell in returning carbon dioxide to the lungs and buffering the pH of the blood is very, very important, but it is not as important as the oxygen carrying capacity function of the red blood cells. So to protect this essential function, the mechanism controlling the development, production, and normal destruction of red blood cells are fine-tuned to avoid interruption in the oxygen carrying capacity or the oxygen delivery capacity of the red blood cell, even under unfavorable conditions, okay, such as when somebody is losing blood, as in hemorrhage. That capacity of the red blood cell to still deliver oxygen is maintained, very well maintained. Okay, so we're going to look at erythropoiesis in this lecture, the process of formation of these important red blood cells in the body. So as a way of introduction, we say that erythropoiesis occurs in the bone marrow. It is a complex regulated process for maintaining adequate number of erythrocytes in the red blood cells. It's a very clear statement. We are going to still see some of these granulocyte forming units. We're going to see the diagram representing how they contribute to the development of the red blood cells. The granulocyte forming, this colony forming unit gives rise to the earliest identified colony of red blood cells called the burst forming unit erythroid, BFUE. Okay, this burst forming unit produces a large multi-clustered colony that resemble a cluster of grapes containing brightly colored hemoglobin. This is just a description of this cell called the burst forming unit. This colony range from single large cluster to about 16 or more clusters. The burst forming unit contain only a few receptors for erythropoietin. Mark it. The burst forming unit contain only a few receptors for erythropoietin, and their cell cycle activity is not influenced significantly by the presence of exogenous erythropoietin. The burst forming unit, these are some of the characteristics. We've mentioned the colony forming units, and then we mentioned the burst forming unit. We are talking about some of the characteristics of this burst forming units, that they contain only few receptors for erythropoietin. The burst forming unit, under the influence of interleukin-3, monocyte colony stimulating factor, troponin, the KIT ligand, develop into colony forming units, erythroid. Okay, colonies. The colony forming unit has many receptors, erythropoietin receptors, and has an absolute requirement for erythropoietin. That is the colony forming unit, erythroid. Their requirement is different from that burst forming unit. So you need to actually be able to differentiate all these cells with their characteristics. That's why I take time to list some of them. Okay, some of the colony forming units are responsible to low erythropoietin. They do not have the proliferative capacity of the burst forming unit because they are a bit committed to erythroid cell lineage. Okay, The erythropoietin, as we know, serves as a differentiating factor that causes the colony-forming unit to differentiate into promonoblasts, the earliest recognizable erythroid precursor in the bone marrow. When you see the diagram, you appreciate these statements further. Erythropoietin is a lineage-specific glycoprotein produced in the renal peritubular intestinal cell. The erythropoietin is like a hormone that stimulates the colony-forming unit erythroid to continue in red blood cell production or maturation. The oxygen, in addition, a small amount of erythropoietin is also produced by the liver. Okay, it's produced in the kidney peritubular intestinal cells, and also a little amount is also produced by the liver. Okay, the oxygen availability in the kidney is the stimulus that activate production of this erythropoietin. Erythropoietin exerts its effect by binding to transmembrane receptor expressed by the erythroid progenitor and the precursor. This is how the erythropoietin exerts its effect on the erythroid progenitor and precursor cells. The erythropoietin also serves to recruit colony-forming units, erythroid, from the more primitive bus-forming units components and prevents apoptosis of the erythroid progenitor and induces hemoglobin synthesis. So this hormone that is produced from the kidney and some liver functions to activate or activate the cell line committed to the red blood cell production to be able to continue in the process of red blood cells. So there are some terminologies we're going to look at, which is going to help us in our description of some of the cells as we go on. We have, the red blood cells. All of us know the red blood cells. They are called the erythrocytes. Okay, the nucleated precursors in the bone marrow are called erythroblasts. So you should be able to also find terminology that is used to describe whether the mature cell or the immature cell. So the red blood cell, is the fully mature cells in the peripheral blood, and they are called erythrocytes. Then the nucleated precursors, the immature ones of these red blood cells in the bone marrow, are called erythroblasts. They also may be called normoblasts, which refers to developing nucleated cells, that is, blasts with normal, some school of thought that call them normoblasts, because although they are still in their precursor stage, development stage, but they are normal. So some people prefer to use the term normoblast. The others use erythroblasts. Okay. This is in contrast to the abnormal appearance of the developing nucleated cells in megaloblastic anemia, in which the erythroblasts are called megaloblasts because of their large size. So when you see megaloblast, it's an abnormal precursor of the erythrocytes, as we see in some megaloblastic anemia. There are three nomenclatures are used for naming the erythroid precursors. Three nomenclatures. The erythroblast terminology, mainly used in Europe. Okay, also, like the normoblastic, we have the normoblastic terminology. It's used often in the US. It has the advantage of being descriptive of the appearance of the cells. Okay, some prefer to use, as we can see in table 1, the rubiblastic terminology, because it parallels the nomenclature used for granulocyte development. So, but the more common one is the normoblastic terminology. As you can see in this table 1, erythroid precursor nomenclature system, you can see the normoblastic, the rubiblastic, and the erythroblastic. So from the primitive immature cell, using this terminology, normoblastic terminology, you have pronormoblasts. Then here we have pro-erythroblasts. This is not often used, this rubriblastic stuff. So we are going to be interchanging between the normoblastic terminology and the erythroblastic terminology. The important thing for you to know is that pro-normoblast is the same thing as pro-erythroblast, depending on the terminology you are adopting or that you are using. We have also the basophilic normoblast, which is basophilic erythroblast here. We have the polychromatic or polychromatophilic normoblast, which is polychromatic erythroblasts. Okay. Autochromatic normoblasts, we have the autochromatic erythroblasts. These two terminology, normoblastic and erythroblastic, are more common, so we are going to ignore this middle rubiblastic stuff. So polychromatic erythrocytes, you can see that at but okay, from this polychromatic stuff, the normoblastic system has gone to erythroblasts. They're almost the same. Erythrocytes, erythrocytes, erythrocytes. So these are some of the terminologies that are used to describe different stages of the normal red blood cell. Remember, we are talking about normal red blood cell, description of normal red blood cells. Okay. So the maturation process of the erythrocytes, the maturation process of the erythrocytes progenitor cells. We say that the morphologically identifiable erythrocyte precursor develop from two functionally identifiable progenitors. You have to understand this very well. Okay, the morphologically identified erythrocyte precursor develop from two functionally identifiable progenitors, the burst-forming unit erythroid and the colony-forming unit erythroid. Both of these cells are committed to the erythroid cell line. That is the most primitive form of cells that can be identified as forming or committed to the formation of erythrocytes. Now, it takes about 1 week for the burst-forming unit erythroid to mature to the colony-forming unit erythroid, and another week for the colony-forming unit erythroid to become a pronormoblast, which is the first morphologically identifiable red blood cell precursor, the pronormoblast. So while at the colony forming unit stage, the cell completes approximately 3 to 5 divisions before maturing further. It takes, we will see this in the next diagram, it takes approximately another 6 to 7 days for the precursors to become mature enough to enter circulation. Approximately 18 to 21 days are required to produce a mature red blood cells from the burst forming the most primitive form of recognizable form of erythroid series. It takes 18 to 21 days. And when you look at the lifespan of the red blood cells, it's within this range. So by the time the one that is already produced is being mopped up from the system, another one have entered the system. Okay, the blood circulation system. So the erythroid precursors, the normoblastic proliferation is a process encompassing or similar, is a process encompassing the replication, that is the division, to increase cell numbers and development from immature to mature cell lineage. I wish I can show the diagram and parallel, but I think it's still down the slides. Maybe we will use the diagram to do that so that I don't repeat. So the normoblastic proliferation, similar to the proliferation of other cell lines, is a process encompassing replication to increase cell numbers. You can see from one cell, you can see the line of divisions we have to increase cell numbers and development from immature to mature cell stages. The earliest morphological recognizable erythrocyte, the pronormoblast, as you can see it here. Pronormoblast derived from the burst-forming unit erythroid and the colony-forming unit potent stem cell. Remember, we said the pluripotent stem cell is capable of self-renewal, is capable of differentiating into different types. So this is one of those different types. The burst-forming unit erythroid is coming from the pluripotent stem cell, as we discussed earlier when we're talking about hematopoiesis. We mentioned about the pluripotent stem cell. So the burst-forming unit erythroid is coming from that pluripotent stem cells. It goes down to form this colony-forming unit erythroid. Okay, as we discussed earlier. The pronormoblast is able to divide. You can see the pronormoblast here. These two pronormoblasts that are able to do what? Able to divide with each daughter cell maturing to the next stage of development. That is the basophilic normoblast, as we can also see in the diagram. So each of these cells can divide with each of its daughter cells maturing to the next stage. That is the polychromatic normoblasts. As you can see it here, they can still divide. These cells can still divide and also mature to the next one. Then also each of these cells can also divide and mature. That is each of the polychromatic normoblasts. They can still divide. You can see it. The prochromatic normoblasts can still divide and still mature. Okay. Okay, so that we can see that in the erythroid cell line, there are many, 3 and occasional as many as 5, there are many divisions of cells with subsequent nuclear and cytoplasmic maturation of the daughter cells. You can see it here. So from a single pronormoblast, which we can, the single pronormoblast, we have up to 8 to 32 mature red blood cell usually results. The condition under which the number of division can be increased or reduced depends on the prevailing circumstances of the body. So let's, if you can see now, from here we have cell division. This cell divided to form this one. There was cell division here, divided to form this one. There are cell division at this basophilic normoblast stage. At the pronormoblastic phase, we still have some cell division. Then autonormoblastic, we still have some autochromatic, we still have some division. But at the end of this thing, you can see that division ends. Division of the cell ends, and what we have now is maturation. Each of these cells begin to mature. You can see the division of the cells have ended, and each of these cells, you can see them, they are now undergoing maturation to mature red blood cells. So that is the process of it. So we are going to look at some criteria in order to identify these cells, whether the mature one or the immature cells. There are some criteria you look out for. Okay, you soon start, I don't know whether I've started staining blood films. Okay, when we stain blood films, we are looking at some characteristics of the cell so that we can identify these cells, are they mature or they are immature, How is the cytoplasm? How is the nucleus? So we call it morphologic identification. Okay, it's individuals that does it. Some machines are capable of also doing that, but it's also good to also do your blood smear to confirm what the machine have already identified. So we call it morphologic identification of blood cells, and this depends on a well-stained peripheral blood film or bone marrow. So you have to carefully make your blood film and stain it properly so that you'll be able to identify the structures of the cells you are looking for. So in hematology, a modified Romanowski stain, such as the right or right Jimsa, is commonly used. The stage of maturation of any blood cell is determined by careful examination of the nucleus. How is the nucleus? And how is the cytoplasm? Okay, some of the qualities of greatest importance we use in the identification of these cells include the nuclear chromatin pattern. That is, when you look at the nucleus, how is the pattern of the chromatin? The texture, look at the texture, how is the density, how is homogeneity. These things, you understand this more by practice. We also look at the nuclear diameter, okay? The nucleus and cytoplasm, what is the ratio? Which one is bigger? The nucleus, how does it compare to the cytoplasm, the size? So we also look at the nucleus and cytoplasm ratio. Then we also look at the presence or absence of nucleoli. Okay, these are some of the characteristics we use to identify red blood cells. And then we look at the cytoplasmic color, the color of the cytoplasm of the red blood cells. So nucleus to cytoplasmic ratio in identifying red blood cells. The nucleus to cytoplasmic ratio, it is a morphologic feature used to identify and stage red blood cell and white blood cell precursors. It can also be applicable. It's also applicable in white blood cells also. So the ratio of the nucleus to the cytoplasm. Okay, this ratio is a visual estimate of what area of the cell is occupied by the nucleus compared with the cytoplasm. If the areas of each are approximately equal, then the NC ratio is 1:1. If the ratio if the nucleus takes up less than 50% of the area of the cell, the proportion of nucleus is lower and the ratio is lower. It is 1:5 or less than 1. That is, if the nucleus is taking a smaller portion of the cell size, okay, the whole cell size, if the nucleus is occupying less than 50% of the area of the cell, then the proportion of the nucleus is lower. Of course, it occupies less size, so its proportion is lower. Then the ratio is lower, okay? If the nucleus take more than 50% of the cell area, the ratio is higher. For example, 3:1 or 3. So in the red cell line, the proportion of nucleus shrinks as the cell mature and the cytoplasm increases proportionately, although the overall cell diameter grows smaller. In short, the nucleus to cytoplasmic ratio decreases as red blood cell matures. As red blood cell mature also, several general trends affect their appearance. We will see that in figure 2. So when you look at this figure 2, we have the A, we have B line, we have the C, then we have the D. So the overall diameter of the cell decreases. See it, starting from the immature, as it's maturing to red blood cell, the overall diameter has decreased in overall diameter of the cell. The diameter of the nucleus decreases more rapidly than does the size of the cell. As a result, the NC ratio also decreases. So you can see this, that the diameter of the nucleus decreases more. The nuclear chromatin pattern become coarse. You can see it. The coarse, as its cell is maturing, the nuclear chromatin is becoming more coarse, clumped, and condensed. Okay, the nucleoli disappear. Okay, at a certain stage, you can see the nucleoli have disappeared. From here, you can no longer see the nucleoli. And these nucleoli represent areas where the ribosome are formed and are seen early in cell development as cells begin actively synthesizing proteins. The cytoplasm changes from the blue-gray or gray-blue, okay, see the color of the cytoplasm changes from the gray-blue to salmon pink. We call the color of the red cell salmon pink. You can see that here. Blueness or basophilia is due to acidic components that attract the basic stain, such as the methylene blue. The degree of cytoplasmic basophilia correlates with the amount of ribosomal RNA. So this is the trend in maturation process of the red blood cell. Okay. So that is the nuclear to chromatin ratio. Another maturation sequence. The sequence changes in cellular diameter, RNA synthesis, and the contents of DNA, the RNA synthesis and content, DNA synthesis and content, that is DNA, sorry, that's a mistake. This is DNA, not banana, is DNA synthesis and content, protein and hemoglobin content during red blood cell development. That is the maturation sequence. When you look at this diagram, you can see the area A, B, C, D. So as red blood cells mature, what is happening? We say that A, okay, because this write-up is trying to describe the diagram, this figure 3. A, red blood cell diameter, you can see this is a graphical description of what is happening during the process of red blood cell maturation. Red blood cell diameter, which are represented in solid lines, okay, they begin to shrink. You can see they begin to shrink. Red blood cell diameter, solid line, they shrink from the pronomoblastic. This is the pronomoblastic. The red blood cell diameter begins to shrink from this stage of the cell to the reticulocyte stage. You can see the reticulocytes here. The red blood cell diameter, that the diagram is going down, so it's shrinking from this stage of the cell, pronomoblastic stage, to the erythrocyte stage. The red blood cell diameter is going shrinking. Okay. The B, RNA, these are maturation sequences. Red blood cell diameter shrinking. RNA, rate of RNA synthesis. What happens to rate of RNA synthesis as the red blood cell begins to mature? We find out that the rate of RNA synthesis, okay, for protein production, of course, is at its peak at the pronomoblastic stage and ends in autonomoblastic stage. So the rate of RNA synthesis in this sequence ends at the autochromatic stage. It's represented in the solid line of this stage. Okay, so what happens, rate of DNA synthesis? You say that the rate of DNA synthesis, also representing solid line here, the rate of DNA synthesis correlates to those stages of development that are able to divide the pronomoblastic, basophilic, nomoblastic, and early polychromatic normoblastic stage. DNA content, dashed line. So rate of DNA synthesis are applicable to the cells that are dividing, and you can see where it stops, where it ends. Okay. At the autochromatic stage of the cell, if you go to the diagram, the autochromatic cell, they don't divide again, they continue to mature. So you say, what happens to the hemoglobin concentration as red blood cell mature, as represented by this box D? Okay, the dashed line represents the total protein concentration, which declines slightly during maturation. Okay, during maturation, declines protein synthesis, which declines during maturation. Proteins other than hemoglobin predominate in the early stages. The hemoglobin concentration, the solid line, can see the hemoglobin concentration here represented with this solid line. Okay, the hemoglobin concentration begins to rise in the basophilic normoblastic stage, reaching its peak in reticulocyte and representing most of the protein in more mature cells. You can see the hemoglobin here, the synthesis of hemoglobin synthesis, okay, begins to rise from the basophilic normoblast. The basophilic normoblast is here. The synthesis of hemoglobin begins to rise from here. Okay, so that is what this graph is trying to tell us. What happens to average cell diameter during these stages of red blood cell development? What happens to rate of RNA synthesis during these stages of red blood cell maturation? What happens to rate of DNA synthesis during the stages of red blood cell maturation? How does hemoglobin concentration behave during the process of red blood cell maturation? So the normoblastic series, a summary of the morphology, you can see these are where the cells are described. The pronormoblast, the diameter is 12 to 20 micrometers. What is the diameter of this? What is the diameter of the basophilic normoblast? The polychromatic normoblast, what is the diameter? Autochromatic normoblast, bone marrow polychromatic erythrocytes, what is the diameter? The nucleus to cytoplasm ratio, you can see this has the highest. As the nucleus is shrinking, this ratio is also decreasing. You can see it, 8 to 1, 6 to 1, 4 to 1, 1 to 2, no nucleus again at this stage of mature red blood cells. Then the nucleoli, you can see has nucleoli 1 to 2, 1. From here, the nucleoli have been extruded from the cell. Percentage of these blood immature cells in the bone marrow, the pronormoblasts 1 to 4% for the basophilic normoblast, 10 to 20% for the polychromatic normoblast, 5 to 10% for the autochromatic normoblast, and 1% of mature erythrocytes, because the erythrocyte, of course, has to leave to the peripheral blood system. So bone marrow transit time, what time does it take the cells to move from the bone marrow to the peripheral blood system? That is what is represented in that section of the table. So we are going to look briefly at these uh, erythroid cell lines, characterize them briefly. Okay, so we start from the most immature erythrocyte cells, which we call the pronormoblastic cell. Its nucleus, how does its nucleus? Is the diagram here is representing the image of the pronormoblastic cell as seen under the microscope image A, and as seen under the electron micrograph in image B. So the nucleus takes up much of the cell. As we said earlier, the nucleus is round to oval. You can see the nucleus here. It's round to oval, containing one or two nucleoli. The purple-red chromatin is open and contains few, if any, fine clumps. So this is a description of the nucleus of this pronormoblastic normoblast. Then the cytoplasm is dark blue because of the concentration of ribosome. This is the cytoplasm that is surrounding the nucleus of the cell. Okay, it is blue because of the concentration of ribosomes. Okay, the Golgi complex may be visible next to the nucleus as a pale unstained area, maybe these areas. Okay, pronormoblast may show small tufts of irregular cytoplasm and the periphery of the along the periphery of the membrane. How does this cell divide? The pronormoblast undergoes mitosis and gives rise to two daughter pronormoblasts, as we saw in the early diagram. More than one division is possible before maturation into basophilic normoblast. Where are these cells located? The pronormoblast is present only in the bone marrow in healthy state. It is supposed to be present only in bone marrow. What are the activity of these cells? The pronormoblast begins to accumulate the components necessary for hemoglobin production, the proteins and enzymes necessary for ion intake and protoporphyrin synthesis are produced. Okay, globin production also begins at this stage. The length of time this stage can last for approximately 24 hours. So we are going to take these cells. Personally, you're going to take these cells, read them one by one, know their specific characteristics. What is specific to this cell that is not applicable to the next cell in the line? You have to be able to differentiate each cell appropriately. Okay, so we have the next cell in the line, the basophilic normoblast. Okay, as you can see also in the microscopic diagram and the electron micrograph of the basophilic normoblast. The nucleus, the chromatin begins to condense. The chromatin begins to condense. This is the nucleus. I've showed you the nucleus already. The nucleus, the chromatin begins to condense, revealing clumps along the periphery of the nuclear membrane and a few in the interior. As the chromatin condenses, the parachromatin area becomes larger and sharper, and the NC ratio decreases to about 6:1. The chromatin stains deep purple red. Nucleoli may be present early in the stage but disappear later. If you look at the cytoplasm, when stained, the cytoplasm may be a deeper, richer blue than in the pronormoblastic stage, hence undergoes mitosis, giving rise to two daughter cells. More than one division is also possible here before the daughter cells mature into prochromatic normoblast. This cells is located only in the bone marrow in healthy state. The cellular activity of this cell is detectable. There are detectable hemoglobin synthesis at this point, but the many cytoplasmic organelles, including ribosomes and a substantial amount of messenger nucleic acid, completely mask the minute amount of hemoglobin pigmentation. It also lasts for 24 hours. This stage in maturation process of the red blood cells. We also have the polychromatic normoblasts, polychromatic, many colors, polychromatic from the name. So the nucleus of this cell, the chromatin pattern varies during this stage of development, showing some openness, as you can also see from the electron micrograph of this cell, showing some openness early in the stage, but becoming condensed by the end. The condensation of the chromatin reduces the diameter of the nucleus considerably, so that the NC ratio also reduces. Okay. And at the end of this stage, no nucleoli are present. The cytoplasm, in cytoplasm, this is the first stage in which the pink color associated with stained hemoglobin can be seen. The stained color reflects over time and concurrent decreasing amount of RNA. The color produced is a mixture of pink and blue, resulting in a murky gray blue. So the pink color of the hemoglobin begins to appear at this stage. Can this cell divide? This is the last stage in which the cell is capable of undergoing division, mitosis. Although likely only early in the stage, the polychromatic normoblast goes through mitosis, producing daughter cells that mature and develop into orthochromatic normoblasts. So this is the last stage where the developmental in the developmental stage of the red blood cell, where cell division, mitosis, can actually occur. It is located only in the bone marrow in healthy states. Why we're emphasizing healthy state? Because in some leukemias, okay, in some leukemia where there is abnormal production of red blood cell, immature cells can leave the bone marrow and appear in the peripheral blood system. Okay, these stages are not occurring in healthy individuals. That is why we are emphasizing in healthy state can only be seen in, supposed to be seen only in the bone marrow. So the cellular activity occurring at this stage include hemoglobin synthesis. It increases and the accumulation begins to be visible in the color of the cytoplasm. Cellular organelles are still present, particularly the ribosomes, which contributes a blue aspect of the cytoplasm. The progressive condensation of the nucleus and disappearance of the nucleoli are evidence of progressive decline in transcription of deoxyribonucleic acid, and the length of time is approximately 30 hours at this stage. So we have the autonomoblastic cells, as we can also see in this figure, the microscopic examination and the micrograph picture image of the orthochromatic normoblast. The nucleus here is completely condensed, which we call pycnotic, or nearly so, as a result of the reduction in the NC ratio is very, very low now. Can see that the nucleus is completely condensed, pycnotic. Okay, the cytoplasm is salmon pink. The cytoplasm is salmon pink in color, and it reflects the complete production of hemoglobin. The residual ribosome reacts with the basic component of the stain and contributes a slightly bluish hue to the cell, but that fades away towards the end of the stage as the organelles are degraded. So what can these cells divide? The autochromatic normoblast is not capable of division due to the condensation of the chromatin. The autochromatic only in the bone marrow in healthy states. The cellular activity going on at this stage is the production of what? Hemoglobin. It continues on, and the remaining ribosomes using the messenger RNA produced earlier. Okay, late in this stage, the nucleus is ejected from the cell, as you can see in the picture where the thing is almost at the periphery of the cytoplasm. The enveloped extruded nucleus, called a perinocyte, is then engulfed by the bone marrow macrophages. So as the nucleus are being extruded from the cell, it's being engulfed by the macrophages in the bone marrow. Often small fragments of the nucleus are left behind if the projection is pinched off before the entire nucleus is enveloped. These fragments are called Howell-Jolly bodies when seen in peripheral blood cells and are typically removed from the cells by the splenic macrophages splitting process once the cell enters the circulation. So this is some of the things we use to describe red blood cell if they are present in the red blood cells. It means that as the process of the extruding the nucleus of the red cells was occurring, it has not completely taken place, but the macrophage have already engulfed part of it that have come out. So the remaining parts can be there, and they are called these Howell-Jolly bodies. The length of time for this stage is approximately 48 hours. Then we have the polychromatic erythrocytes or the reticulocytes. The reticulocytes, as you can see in this diagram, the nucleus is, you can see, this is the beginning at the polychromatic erythrocyte stage. There is no nucleus. You can see these cells now. There is no nucleus. The polychromatic erythrocyte is a good example of the prior statements that a cell may not have all the classic feature described, but may be staged by the preponderance of features. And particularly when a cell loses its nucleus, regardless of cytoplasmic appearance, it is a polychromatic erythrocyte. The cytoplasm can be compared with that of the late autochromatic normoblasts in that the predominant color is that of the hemoglobin. So by the end of the polychromatic erythrocyte stage, the cell is the same color as a mature RBC, that is salmon pink. It remains larger than a mature red cells. It remains larger than a mature red blood cells. The shape of the cell is not the mature biconcave disc. You can see this shape on this electron micrograph picture. It's not that of a biconcave disc of the mature red blood cell. You can see how the shape of the erythrocyte is under the electron micrograph, but it is irregular in an electron micrograph. Select nucleus, of course, the polychromatic erythrocyte also cannot divide. Location: this erythrocyte resides in the bone marrow for a day or longer and then moves into the peripheral blood for about a day before reaching maturity. So the cellular activities that are also happening in this erythrocyte stage of red cell maturation is that the polychromatic erythrocyte completes the production of hemoglobin from residual messenger RNA using the remaining ribosomes. The cytoplasmic protein production machinery is simultaneously being dismantled. Ribonuclease in particular digests the ribosome. The acidic component that attracts the basophilic stain decline during this stage to the point that the polychromatophilia is not readily evident in the polychromatic erythrocyte on a normal peripheral blood film stained with right stain. A small amount of residual ribosome is present, however, can be visualized with a vital stain, such as the methylene blue. Okay, That is how we can stain to be able to identify the erythrocytes using these vital stains. That is staining the cells while they are actively, while they are active, okay? The residual ribosome appear as a mesh, small blue strand. You can see the small blue strand residual inside of the cell. That is how we can identify these cells as a reticulocytes. You can see the mature red cells are beside it without these residual ribosomes on inside it. But here, this is how we can identify cells as these red cells are able to. Okay. So how the name reticulocyte is often used to refer to the stage immediately preceding the mature erythrocytes, even when stained with ricin and without demonstrating the reticulum. Those that are joining the class now, I'm not supposed to admit you. So the length of time, okay, the cell typically remains a polychromatic erythrocyte for with the first 3 days spent in the bone marrow and the third spent in the peripheral blood, although possibly sequestered in the spleen. So we're going to look at the erythrocytes. We're going to look at the erythrocyte, which is the mature red blood cell, the final stage in the maturation of the red cells. Again, you can see in the figures the microscopic image of the red blood cells. Can see it here. This area that we call them area of central pallor. Can see them here. Can see the diagram of the same erythrocytes under the electron micrograph image of the erythrocytes in this B part of the image. So the nucleus of the mature red blood cell, the erythrocytes, there is no nucleus. The cytoplasm, the mature circulating erythrocyte, is a biconcave disc measuring 7 to 8 micrometers in diameter with a thickness of about 1.5 to 2.5. Okay, on a stained blood film, it appears as a salmon pink. So you can see here, it appears as a salmon pink staining cell with a central pale area that corresponds to the concavity of the cell. The central pallor is about 1/3 the diameter of the cell. So this central pallor is supposed to take about 1/3 of the diameter of the cells. It also helps us that observing a stained film, a stained peripheral blood film, if the area of the central pallor is more than that, it can be an indication of anemia, that the hemoglobin content of that cell may be low. So when we are looking at the blood stained film, you check the area of the central pallor. It's not supposed to be 1/3 of the 1/3 of the area of the cell. Okay. The erythrocyte cannot divide. Okay. And then location and length of time active on the peripheral blood for approximately 120 days. Again, leads to their removal by the spleen. So what's the cellular activity? Okay. Of course, you should be able to describe that on your own. The mature erythrocyte delivers oxygen to tissues, release it, and returns to the lung to be reoxygenated. The interior of the erythrocyte contains mostly hemoglobin, the oxygen-carrying component. It has a surface to volume ratio and shape that enable optimal gas exchange to occur. with the biconcave shape, even hemoglobin molecules that are towards the center of the cell are not distanced from membrane and are able to exchange oxygen because of the shape, the biconcave shape of the cell. Oxygen exchange is very, very easy and effective and efficient. The cell's main function of oxygen delivery throughout the body requires a membrane that is flexible and deformable, that is able to move within either smaller vessels, even the capillaries move inside tiny blood vessels. So it needs to be flexible to be able to change and be able to maneuver its ways and move around even to the tiniest capillaries to be able to deliver oxygen to very important body organs. The red blood cells must squeeze. I've said that. Okay, deformability is crucial also in that the interaction of various membrane components creates the properties of this flexibility of the red blood cells. So this is where we're going to end today. We've been able to characterize the red blood cell, be able to describe by describing the various stages of red blood cells from the pronormoblast to the basophilic normoblast to the polychromatophilic normoblast to the autochromatophilic normoblast, reticulocytes and erythrocytes by describing the nucleus, the cytoplasm, the cellular activity, the length of days. We are actually characterizing these cells for you to be able to identify them. So you have to go back and look at these cells, stages of maturation of these cells, and be able to differentiate them both in their morphology and in their functions and otherwise. Okay, thank you for listening. We're going to stop here. Next time, we're going to look at we're going to stop next time. We are going to look at white blood cell leukopoiesis of white blood cells. Okay, so if there are any questions,